Okay, I wanted to go back to stream succession for just a minute because I was looking through and there is a figure that we can use. Here's the color version of the number of larvae per sample of perch and crappie and pumpkin seed and bluegill that we talked about. So I have an equivalent one of the kinds of trends that you might see in the stream. And this particular stream is Sycamore Creek, which is a desert stream in Arizona. And this is a severe flood comes in, and you have almost 90% cover by just bare sand and 10% covered by diatoms right after the flood. But then as it calms down, the diatoms really take off and bloom. And slowly, though, they go down, and other things that are better competitors under more stable conditions take over. Eventually, the cyanobacteria take over. So this is just a contrast, the kind of thing you might see. We see this at Kanza every single year. We have the drying and the flooding. And so we have this series of events where the algae come back first. The fishes, the herbivorous fishes and such can come in pretty fast, and the invertebrates can reproduce. Once they can reproduce, they'll come up. And we see that changing the system. And if you have a severe flood, then you also have a sequence that happens, although everything's connected, so things come in faster. So we're talking about indirect interactions when we left off the last time. And I gave an example of Chlorophyra. And I just wanted to give a little bit of theoretical background to give you all an idea of how complex things really are. So if we think about just 7 species, and we can see that the number of direct interactions between those is, the total number is 7 species minus 6, right? Times 7 minus 1, 6. That's because each species interacts with 6 other species, right? And then you go to the second species and interacts with 6 others. So that's all the two-way interactions. And so that's 42. But if we have 7 species and we think about the indirect interactions, right? But it's going to end up, the equation is actually, there's the 7 times 6, right? But then you get 5 more for, times 5 times that for the 2-link interactions, right? Not the single direct interactions, but the first link of indirect interactions. 4 times as many, 3 times as many, 2 times as many, and then just 1 time for this, the 7, the full 7 linkages in that. Okay, so that means is, if we look at the number of species, a number of links and possible chains, we just plot that out, right? The number of direct interactions is down here, but with just 7 species, we're up to over 50,000 links of 5 of length 5 and 50,000 of length 6. So include pathways that include 5 species. Right, so this is not even close to what a community really is, right? I mean, it's just 7 species. And so the question is, how do you weight these indirect interactions, and how do you deal with that complexity? It's been one of the big issues in ecology, and you know, it's difficult to make ecological predictions based on that. We talked about that already. So this is just a graph of that kind of thing. And this is a food web from Little Rock Lake. And it's, the bottom is the food sources, which are probably not resolved as well as they should be. These here are not resolved as well as could be. They didn't go like to every species of algae, for example, because it's a planktonic food web. Then the primary consumers, things like zooplankton and such, and maybe things that eat the zooplankton. And finally, there's the fishes are up here, up in the secondary or tertiary consumers. One thing that you can notice here is that some of these, so these each line is an interaction with, you know, in this case, a food web, and these things are eating that. One of the questions I'll have for you is, what does this line mean? So there's a line here that connects a node to itself. So that could be a big bass or something. I don't know, some big fish at the top of the food web. And there, if you look actually closely, there's a number of those where there's these loops. Yeah, Cassie got it, right? You eat your own. A lot of big fishes just eat little fish, and they don't care if they're their own, you know, offspring or somebody else's offspring that's the same species. So that's good cannibalism. Right, so that adds another level of complexity to all this. And if you were to model this and say, okay, I'm going to increase temperature with global warming, what's going to happen? I don't know. And, you know, will a trophic cascade work? You know, because some of these links are not very set on a, it looks like there's 2 trophic levels, maybe 3 here, but then some of them are sort of up a half again still. So that's another issue to think about. One of the real hot topics for management of ecological systems is the idea of thresholds and breakpoints. I'm going to talk about a few of those. And I should mention that this is not on, I'm going to have to reload my slides because I decided I wanted to add this in just like this morning. And so I pulled some stuff from other places. So what I'll do is I'll upload the slides, re-upload the slides, and then you can get them if you want. There's a few other things that we've changed over time too, so I might as well just go ahead and do that. Just this last batch on the Canvas site. So there's a threshold where if you push a system with an independent variable or a driver, we call them sometimes, you know, not much change, and then all of a sudden you push them just a little bit more, and there's a big change. And so that would be a threshold or a breakpoint. If that threshold or breakpoint, pushing that system over that threshold makes it so it can't recover quickly, right? It's like if you pull back on that, you know, if it just falls back, it's resilient, right? And then but if it doesn't fall back, there's hysteresis. If it takes longer to come back than it took to get there. So that's the idea of an alternative stable state. And this is kind of what we want to avoid. We want to know how hard to push systems. And I should define 2 more terms here, which we've already talked about a little bit, resilience and resistance. So systems can be either resist change, resistance, right? Or they can change a lot, but then pop back to where they are. So they can be quite resilient. So like we have an example of a grassland that burns, right? It just pops right back, even though it changes a lot for a while, it grows all right back. Or a flood where the algae grow back really, really fast, as opposed to the fish, which dive down in the sediments to get away or swim to the edges. They're resistant to floods, right? They're not resilient because they can't reproduce as quickly. Okay, Emma asking, what way are you pushing a system? Well, yeah, so that could be anything, right? You could be changing the temperature, you could be increasing the amount of nutrients, you could be adding an invasive species, anything that drives an ecological response, right? And then one of the questions is, how much of a change does there have to be before you're in a new state, right? Officially in a new state, and that's a fuzzy area. Not state of the nation, but ecological state of being. Okay, so we've talked about some things already that are really good thresholds, really clear thresholds with an alternative stable state. And this is the one, and I actually, after we were talking, I regraphed this because we had the oligotrophic lake and the eutrophic lake. So now I made the oligotrophic lake blue and the eutrophic lake green. And if in this case, you're pushing by adding, eutrophication is pushing by adding more and more phosphorus. And at some point, the algal bloom on the top sinking down into the hypolimnion causes the entire hypolimnion to go anoxic, right? And so the iron and phosphorus that would be precipitating out and being stored in the sediments in the oligotrophic system are redissociating, and then in fall mixing they can come back up and refertilize the whole system. And it's called internal loading. So we have 2 distinct biogeochemical states here, right? One where the iron and the phosphorus are trapped in the sediment in the oxidized microzone here when the oxygen's in the hypolimnion, and the other here. So a very clear threshold. And it's a alternative stable state, and there is hysteresis, because if you fertilize to the point where you get this happening, the phosphorus keeps coming back up again, and you got to get it out. Anybody remember how they did that in Lake Truman when we talked about the eutrophication of Lake Truman and how they fixed that problem there? That was a Swedish lake, I believe. Nobody's answering. Took out the top layer, so a good Mac. Yeah, they dredged it. So they dredged all that high phosphorus stuff. So it was like this, and there's a bunch of phosphorus in the sediment. They just scooped it all out and made it deeper. So yeah, so that's a big amount of work to push the system back to a desirable state, right? So that suggests they really had to push to get it back into the desired state. Here's an example of something that you could call a threshold, but I don't really necessarily think there's a threshold or a breakpoint per se, because if you look at this on the left, so this is the ammonium concentration, and this is phytoplankton uptake rates of ammonium. So this is the uptake rate. And we see that we have a Michaelis-Menten relationship here, right? Here's the mathematical description, and we talked about this back in chapter 17, I believe, or something like that. And we see that we can fit it pretty well with this Michaelis-Menten curve. But if I go and zoom in, like I would say, looking at this, say, oh, well, look, there's a threshold at about 2.5, right? That it goes up and then boom, it just doesn't anymore. But if you actually look at the mathematics of this and zoom in between 1 and 4, it's still a curve that's going up smoothly, right? So this is one also where that if you took the nutrient away, it would just come right back to the nutrient uptake rate. It'd come right back probably to what it was with the low nutrient before, suggesting it's not an alternative stable state and it's not really a sharp threshold. So stated differently, there's not one little break point where you go a little bit further and it changes really rapidly. What's happening is that it's changing really rapidly here and then just a little bit less, a little bit less, a little bit less rapidly going on. So that's what you could say, look, there's a break point, right? You could put 2 lines on there, but if you go close, you're not going to see that. Okay, so we can also talk about what is happening in the Ogallala Aquifer and also in western Kansas is another threshold break point. So we see that the first big change that happened to the watersheds in this part of the state to the west are agriculture. Row crop agriculture led to a lot of siltation. We had 2 really big droughts in the 30s where the Dust Bowl was, right, in the 50s, which was almost as intense of a drought, but it didn't got this dust going in the soil. Center pivot irrigation started in the 60s, so pumping up water from the groundwater to irrigate fields. And then a lot of reservoirs were built between the late 40s and the early 1970s. And what this has done, it's made a new threshold. And this is an example then of discharge of one of the rivers in this area. And we see that while it's quite variable, boom, 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 up here, when we get to the center pivot and the reservoirs. So why would reservoirs decrease the flow overall in a stream or in a river? They hold back weather, but for a while, right? But eventually it's got to come out. I mean, you can't just, well, I mean, what's the loss that that's going to cause? Right, like if you shut down Tuttle Creek and held all the water back, eventually it'd just go over the spillway because they couldn't. Oh, good, Matt got it again. He's on a roll today. Evaporation, right? Yeah. So especially in the west where it's really dry, if you get a bunch of surface area, and you'll get a lot of evaporation. So you lose there, And then also with this center pivot irrigation, you're pulling the groundwater out from underneath these streams. And so we've gone to a new state where we have much lower flow, much less intense flooding, right? Even when you get flow, and then eventually it'll be dry. So this is just an example of the Arikaree River, which is one of the rivers in western Kansas. And the first picture is kind of grainy, but you can see there's a big wide flowing river here, right? That sandy bottom river, and probably in the springtime, maybe not all the year. By 1996, the flow decreased enough that the riparian vegetation is starting to move in, and it's narrow. The river's getting narrower and narrower because it's not flooding like it was to just blast out all the sand here. And here's in 2006. There is not a river there anymore. It's a meadow. And so this is the actual the photographic evidence of what's happening in a lot of places in western Kansas related to this. So I'm going to talk about some work that Keith Gitto's done and Josh Perkins done too, on this. And if you're in Keith's class, don't tell him I'm telling you about fish stuff, but it is kind of cool. Okay, so what they did was look at the historic fish collections by going into museum specimens and data. And we see that in 1965, the areas that are purple are the ones that are still flowing. Okay, and so this is just that chunk of western Kansas I was talking about. And the dark ones are dots that were taken, fish collection that were taken pre-1970. So remember that break point, you know, where we saw the big fall off was sort of between 1960 and 1975. And then at those fishes that are sampled afterwards, where they're collecting fishes afterwards. And we do see that almost all of these collections are happening in these purple areas. So where the groundwater keeps the stream flowing in the dry periods, that's the refugia for the fishes. So by 2005, let's toggle, see what happens to the purple. Like look here, down here. So in 2005, 1965, this is all purple and flowing all the time. It's all the way down to here. So now, these places where people were collecting fishes pre-70s and even by 2003 are not good refugia, right? They're not, the fishes are not able to make it. And by 2020, almost all gone, right? So now, this is just a little bit here, purple here. So we go through it again, I mean, we can just pick a spot, like pick this river here and watch what happens over those years. It's just, the flow's gone away. Pick this one here. This one's a little bit better off, right? So this one's a little bit deeper and these are probably gonna make it. Pick this one over here. Pretty low. All right, so this is really tough on these fishes to come in and repopulate areas. What's happening is this is leading to extinction of some species of fishes in western Kansas. And the particular species that are having troubles are the broadcast spawning fishes. And they have eggs that are just slightly less buoyant than floating. They just sink a little bit. So they lay the eggs and put them out in the stream, and then they work their way down the stream, and the turbulence keeps them in the water. And they have to be kept in the water. They'll go down the sediment and smother out. If you don't have places where there's water flowing, long stretches where the water's flowing, you can't pull that off. So this kind of fragmentation has really hurt those species. And you add on to that throwing reservoirs in, because the same problem happens. Even if you just flow into a reservoir, then the water's still, and they go down to the bottom, and that's it for them. So we're losing prairie fish species related to this. This is a threshold in an alternative stable state for two reasons. One, it takes thousands of years to refill the aquifer underneath this all. So even if we immediately stopped pumping water out, it would take thousands of years for that to come back. It takes even more than thousands of years for new species of fishes to to evolve. So we're talking, 40 years here to lose the species and change the hydrology completely, and thousands and hundreds of thousands or more of years for the new fish diversity to come back into the system. So this gets at the point that extinction, we talk about the chytrid disease in the frogs, is a alternative stable state. It's not one you can come back from quickly. Another example of a threshold, and this has implications for water quality protection. We already talked about this earlier in the class, but I want to bring it up again. So if we look at the invertebrate richness versus total phosphorus in streams, and these streams are those found in Missouri and Kansas and Nebraska. And we can see that if the phosphorus is quite low, you can have up to maybe 120 species, 100 species. You may have really low diversity, but you can have over 100 species. But where you have high total phosphorus, you really don't have a lot of species that often. And it turns out there's a threshold here, and you can get this number statistically. And you can put a confidence interval around it of about 0.05 milligrams per liter, which is about 50 micrograms per liter, right where we think that the sort of the reference condition is. Okay, so her hypothesis, Michelle Evans White, who did this work, is that with stoichiometry, food becomes more phosphorus rich with phosphorus pollution. So all the things that the invertebrates are eating are more phosphorus rich: the leaves, the algae. And invertebrates that grow more quickly have a higher phosphorus demand because they need a lot of nucleic acids to make things churn along, right? And given this, she thinks that she thought that the primary consumer community should be the most sensitive, right? So we already talked about carbon to phosphorus ratios. If an invertebrate's eating another invertebrate, they'll have pretty much a balance of carbon and nitrogen and phosphorus that they need. Maybe less carbon than they need. But if you put more phosphorus in, then the specialists that need more phosphorus do better. Predators should not be as sensitive because their food source is relatively phosphorus constant, right? That's what I was trying to say there. So she went in and plotted these, the thresholds that she found versus total phosphorus. So she made graphs just like this, but she did it just for predators, just for grazers, scrapers, shredders, independently. And we see that all these things, the things that eat leaves, the things that scrape biofilms off of rocks, the things that collect, all have lower threshold values than the invertebrate predators. So this lined up with our hypothesis. Is this threshold an alternative state? That we don't know. That if you drop phosphorus back, how quickly everything will come back into the diversity that we'd see at low phosphorus naturally.